Okay, perfect. Thank you very much for having me. I will be talking today about bio-behavioral synchrony and interbrain coherence and attachment perspective. First, I would like to say thanks to everybody who has contributed to this research. It's a growing list of people, and without those people, it wouldn't have been possible. So I'm really, really grateful, and I hope that I can keep up these collaborations in the future. Although this might be a little bit of a repetition, I would like to give you a short introduction into the field where my research is located. And this is mainly within the field of social neuroscience. Social neuroscience has been first introduced in 1992 by John Cacioppo and Gary Berntsen as a novel combination of biology and psychology, two fields that until then had been pretty separate. And during the decade of the brain, John and Gary were proposing that these two fields should be combined so we could actually study what the biological and brain substrates of human social behavior are. Now, even though a big focus has been directed to the brain, as can be seen also with the icon that I have put to this question, social neuroscience actually is a multi-method and multi-level approach. So we are not only looking at the brain, but we are looking at the whole organism, from DNA, from epigenetics, to physiology, to organs, to the whole body, and also to social interactions between people and even to cultural aspects. So it's a vast array of considerations that social neuroscience is investigating. This being said, even though social neuroscience is aiming at looking at how the brain or the body processes social information, very much of it has been focusing on one participant in isolation. What does this mean? It means that we have been using so far different methods, like, for example, functional magnetic resonance imaging, EEG, where we have been showing participants films or movies or playing voices, but they have always been alone while doing that. And even when we consider paradigms like the Trier social stress test, where we are inducing stress in a social setting, we have been only looking at the brain responses and the physiology in the person that is being stressed and not as a function of the interaction that is actually happening. But during the last 5 to 10 years, there has been a very strong transition within the field of social neuroscience, because we have been moving into second-person social neuroscience, where we are looking at direct interactions between two or more participants at the level of the brain, but also at the level of the body and genes and all other levels that can be looked at. And I'm sure that in the first talk today, Leo has already told us why this is the case, namely because social cognition is fundamentally different when we are engaged with others rather than just observing them. So in order to really understand what's going on in the brain and the body when we are interacting with others, we need to look at both people, or ideally at three people or even bigger groups at the same time. Now, this being said, we are of course still measuring individual activation patterns in two people, for example, when they are interacting with each other as depicted here. However, the very nice thing is that we can then look at the temporal alignment or the attunement of activation patterns between those interacting brains, between those interacting partners, to really see how these different types of responses line up with each other. And this is, of course, not only possible for the brain, but we can also do that for any other of these levels that I have been talking about. And in so doing, we are actually moving into the domain of bio-behavioral synchrony. And this domain has been pioneered by Ruth Feldman, who has published a very nice paper on this just 3 years ago. And I'll be referring to this paper now for this slide. And Ruth Feldman says that bio-behavioral synchrony during or immediately after social interactions is actually most observable at 4 levels of the body and the environment. And these 4 levels are depicted here and now framed red. So they include behavior, for example, eye gaze, touch, Then they also include physiology, so we can have synchrony in the heart rate, for example. Then endocrinology, so we can have synchrony in the secretion of cortisol or in the secretion of oxytocin. And finally, in brain activation patterns. Importantly, what Ruth Feldman suggests is that there are different degrees of synchrony as a function of the intimacy or the social closeness of interacting dyads. And she suggests that synchrony should be strongest in most intimate, most close dyads, like, for example, parent-child bonds or romantic partners, and that synchrony should then decrease when we move away, when we move to more distant people, for example, friends, and finally strangers. So she suggests that there are degrees of synchrony as a function of relationship categories. We are suggesting an extension to this view because we think that the degree of biobehavioral synchrony should not only vary between those categories of social relationships, but also within categories as a function of inter-individual differences in relationship quality or attachment. So, for example, we would propose that within parent-child interactions that should have the highest default synchrony because it's the most intimate bond, we should find differences as a function of relationship quality and mother-child attachment. And for the most of my work on this question so far, we have been focusing on brain-to-brain synchrony, so looking at the temporal alignment of brain activation patterns. Why are we looking at brain-to-brain synchrony or interbrain coherence as another word for that? So why is this important and why is this relevant? And as has already been shown in the talks preceding my presentation, the general idea is that for successful social interaction, the oscillatory processes interacting individual brains, they must become synchronized in order for information to flow between them. And this is thought as analogous to a wireless communication system, right, in which two brains are coupled by the transmission of a physical signal through the shared physical environment. Now, this might sound a little bit like telepathy, where we are trying to invent fancy machines to mind read and to infer the mental states of others, as depicted here in this father-child dyad. But of course, it's no magical kind of telepathy thing, but it's rather, as Sam has, for example, shown just an hour ago, that we are needing some cues in the environment that face lock our brain activation to the person we are interacting with, that we can have the same percepts or the same mental processes at the same time. And some cues that are used for this, for example, are pointing or eye gaze during interactions that really make us attend to the same stimulus at the same time, so we can have synchrony within the brain to certain attended information. Now, how do we measure interbrain coherence? In contrast to what has already been shown before, like EEG, or it can also be done with fMRI or MEG, we are using functional near-infrared spectroscopy, or short FNIRS. FNIRS relies on the same principle as fMRI, so it measures blood flow and it measures blood oxygenation, but it does so with infrared light and not with a strong magnetic field. So there are clear advantages. For example, we have a higher sampling frequency, but we have also a much lower susceptibility to movement artifacts. So we can do this while people are sitting or even standing. There are also, of course, some disadvantages, and these are related to the spatial resolution. So we can only measure brain activity close to the scalp surface. We cannot go to very deep brain areas. But I think this disadvantage is pretty well compensated by the other advantages that I have just mentioned. So, for example, if you imagine now that we are measuring two brain activation patterns using FNIRS like depicted here, one in red and one in blue, we can then plot these into the frequency by time domain. So frequency here is depicted on the y-axis and time on the x-axis. And as you can see in both participants, we have a frequency band that we usually relate to the heartbeat. So we can clearly see the heartbeat frequency in our data. And this is also usually a very good quality check. So if we don't see that, then probably the channel is not so good. But what is of more interest then is the frequency that is related to the task. And here depicted is a frequency that was related to button press. And you can see this in both participant 1 and participant 2, independently of each other. What we then do, and what is the most crucial step in Effner's coherence analysis, is that we correlate those signals for each frequency band, and that you can see now here in panel D. And this can also lead to some synchrony seen in the heartbeat, but since the temporal resolution there is not so good, we usually don't look at that. But we rather focus on the task-related frequency, and here you can see that there are some nice time points where the task-related frequency actually was correlated between participant 1 and participant 2, and we can then take this and run additional analyses on that coherence. What might be important in this context might be to say that we are talking about coherence and not so much about synchrony, because in order for two signals to be correlated with each other, they don't have to be necessarily completely synchronous. So we can also have coherence when the signals are a bit not in the same phase, so when there is a phase shift, or if they are even in opposite phases. The important thing is just that we have a continuous regularity that can be picked up by the correlation. So I would rather refer to coherence than synchrony, but these terms are used both, and they refer to the same construct within this context. Now, the second question is, why would attachment be important for bio-behavioral synchrony, or why would we expect differences in relationship quality to modulate bio-behavioral synchrony? And here we think that this is quite evident because social synchrony is learned within the parent-infant bond, which is the strongest attachment bond, meaning that attachment contexts provide the arena for the experience and encoding of synchrony early in life. And there is also the notion that these early experiences will importantly shape how we experience attachment bonds later in life, and how our body and brain respond to such interactions in terms of bio-behavioral synchrony. And in that regard, we think that attachment theory that was developed by Mary Ainsworth and John Bowlby about 60 to 70 years ago, and has nowadays become the most comprehensive psychology framework of the 20th century, and it's now also still being very prominent in the 21st century. We think that this concept is really nicely suited to study bio-behavioral synchrony, especially because attachment theory puts individual differences in how we form and maintain close relationships throughout life at the very core. So we can really look at how bio-behavioral synchrony varies in different social interactions as a function of relationship quality if we refer to attachment theory. How do we do that? Most importantly, we use video recordings during experiments to and then coding according to attachment theory-based schemes to look at the quality of the interaction as it actually unfolds in real time. We can then also use self-report questionnaires filled in by the adults or by parents of children that participate. We can furthermore use specific narrative tasks or interviews to look into early attachment experiences and how they are mentalized by our participants. And finally, we can associate these measures also with physiological variables, because there is a large amount of literature showing that attachment and physiology are quite tightly linked. Okay, having given all these this theoretical background, we can now move to our studies. And these have started already quite a while ago, 7 to 8 years, when I was still at Stanford University, where we had one paradigm in adult dyads and one in mother-child dyads. I continued this work when I moved to Leipzig, where we set up a very close collaboration with researchers from the University of Vienna, where we had another set of parent-child interaction paradigms, this time both in mothers and fathers. And we are just currently also looking at the new study in romantic couples, where we look at stress-related response. Now these are shared between the dyadic partners, the romantic partners, during the social stress test, Trier social stress test. But for today, I would like to focus on 3 studies that have looked at parent-child interaction, one from Stanford and then two from Leipzig in collaboration with the University of Vienna. And I would like to start with the Stanford study. This study was mainly led by Jonas Miller and was in the group of Alan Rees at the School of Medicine at Stanford University. And in this study, we had 28 mother-child pairs that were brought in the lab for the setup that you see in the picture labeled with A. Unfortunately, I cannot show the mother and the children because of data protection issues, but this was more or less the paradigm or the setup. And the paradigm was a reaction time task where the participants had to press a button as quickly as they could when the circle, an empty circle filled with green color in the independent condition. And they had to try to press the button as simultaneously as possible with the other participant in the cooperation condition. Now, this is a tightly controlled behavioral paradigm, but like Sam has just said before, we could also expect synchrony in such a tightly controlled paradigm. And there were 40 trials for each condition in 2 blocks with rest phases in between. And we measured brain-to-brain synchrony in 5 regions of interest on the skull surface, one at the temporal, in a temporal region overlapping with the temporoparietal junction that is important in mentalizing, theory of mind, and then 4 ROIs in the right prefrontal cortex, also important for mentalizing and emotion regulation. So what did we find? So the first main finding was that when we compared the cooperation condition showed in light gray and the independent condition showed in darker gray, that there was an increase in interbrain synchrony or interbrain coherence during the cooperation condition. And this is in line with other studies from parent-child interaction showing that if we are interacting with somebody, then the brain-to-brain synchrony increases as compared to when we do a very similar task independently of each other. Interestingly, we also found some sex differences in this pattern, in that the difference in interbrain coherence between cooperation and the independent condition was only significant for mother-son dyads, but not for mother-daughter dyads. It's still a bit difficult to point to a specific mechanism behind this sex difference. We think that it might have to do something with the fact that boys might be a bit more competitive than girls, but this is still a very open question. But I think that it's good to keep this in mind for later when we talk about mother-child versus father-child dyads. Finally, what was most important in this study for us was that in one region of the prefrontal cortex, that is pointed out in purple on the left, we found a correlation in the amount of synchrony during cooperation and attachment style from the child towards the mother. And usually in attachment research, we talk about 3 attachment styles, 1 secure and 2 insecure. And in this case, we found this correlation with attachment avoidance, which is usually associated with a strong desire for independence and not really perceiving social interactions as strongly rewarding. And in that sense, this finding is quite well in line with the literature, because it shows that the more the child was avoidantly attached to the mother, so the more it showed a desire for independence and the less it experienced social interactions as positive, the lower the coherence was during the cooperation condition. Now, importantly, so this gives some clue about the relationship between brain activity or interbrain coherence and relationship quality. Importantly, this finding did not survive more stringent correction for multiple comparisons. So we still have to regard this finding as preliminary, but we think it's very interesting because it points to the right direction. When I then moved to Leipzig, and we set up a new study with strong collaboration with the University of Vienna, and we tried here to be little bit more ecologically valid by not having a so strongly behaviorally controlled paradigm. And this study has just been published a couple months ago with first author Jin, who has been very strongly involved in this and who has done a wonderful job. And in this experiment, she recruited 42 mother-child pairs, as you can see in the picture. The children now were younger than the ones in Stanford, so they were 5 to 6 years old as compared to 8 to 12 years old. And the dyads were playing a tangram task, so they had some. They had to, with 7 puzzle pieces, they had to recreate some templates, as you can see now here in the picture. And they either did this together, so they had one puzzle set for both of them, or they did this independently of each other, separate by a removable barrier, and each of them had one puzzle set for themselves. And we had some rest phases in between. And we checked the brain-to-brain synchrony at 2 areas bilaterally, the dorsolateral prefrontal cortex and the temporoparietal junction. So we had slightly different regions of interest as compared to the Stanford study. So what did we find here? Firstly, and most importantly, we saw that across all the 4 regions of interest, there was higher synchrony, higher interbrain coherence during the cooperation condition, showed in red, as compared to both the individual condition, showed in green, and the resting condition, showed in blue. So this was another nice confirmation of the general notion that when parents and children do something together, they have higher interbrain synchrony, interbrain coherence than when they do the same thing independently of each other. Importantly, we also validated this finding with random permutations, where we paired mothers with random children or random children with random mothers, just to make sure that we don't have any spurious correlations. And this effect of increased interbrain coherence was strongest in the right tempororietal junction. We then looked whether there were any correlations between the coherence during cooperation and some of our measures. And we found that, for example, the coherence was higher the more templates the participants solved. And the coherence was also higher the more reciprocal the interaction was. So the more behavioral turns the dyads were taking during cooperation. Furthermore, we found that interbrain coherence during cooperation was higher the more the child was allowed to take the lead rather than follow the parent. And finally, we also found that interbrain coherence during cooperation was lower the more the mother reported to be stressed by her role as a parent. So here we find very nicely that interbrain coherence goes together with behavior, so that it is actually functionally implicated and important for cooperation behavior, and that it also associates with some state traits relating to parental stress in the mother. And both of these variables point to the fact that interbrain coherence is actually related to the interaction quality and the relationship quality between the mothers and their children. In the same participants, we also had another paradigm that we wrote up in a paper that has just been accepted a couple days ago in the journal Scan. And in this paradigm, the mothers and the children were told, instructed to just freely talk to each other for 4 minutes. And we had these data from 40 mother-child pairs. Again, the children were 5 to 6 years old. We behaviorally coded these free verbal conversation on a range of properties, but most relevant for today is the turn-taking property. So we checked whether the dyads actually engaged in reciprocal alternating utterances, or whether they were these the instances of communication were actually not so reciprocal and were not really following each other in a nice turn-taking pattern. And we separated the 4 minutes into 8 epochs of 30 seconds, and we checked whether the brain-to-brain synchrony, the interbrain coherence, changes during those 8 epochs of 30 seconds. So what was our findings? Firstly, we had to do again these random permutations because we didn't have any control conditions, so we had to make sure that our correlations are not spurious. So we checked the real pairings with arbitrary pairings, and we indeed found that real dyads showed significantly more interbrain coherence than random pairs during conversation. Secondly, we found that in the real dyads, interbrain coherence increased on average over time. So the longer the mothers and the children were talking to each other, the more there was interbrain coherence. We then also saw that in real dyads, interbrain coherence was positively associated with turn-taking. So the more utterances were taken by the dyads in a nice reciprocal and turn-taking manner, the higher the interbrain coherence was overall. And finally, most interestingly, we found an interaction between interbrain coherence, time, and turn-taking, in the sense that in dyads that showed overall higher turn-taking, so that were higher turn-takers in general, only in those dyads the interbrain coherence actually increased with time. We didn't find any associations between interbrain coherence and the other measures that we coded from the interaction, like relevance, contingency, and intrusiveness, but I have not talked about those today. So what does this show? This shows that interbrain coherence does not also only associate with behavior that we are exhibiting, for example, during cooperation, during puzzle solving, but also with talking to each other. So it is enough to just freely talk to each other to induce more coherence. And the more actually these utterances are in a turn-taking manner, the higher the coherence is. And we think that this is really an interesting finding relating learning and mother or parent-child interaction in general. And that this, of course, again, gives us some idea about relationship quality, because usually reciprocal interactions are of a higher quality, and reciprocity, for example, also relates to parental sensitivity, which is an important construct in attachment research. Finally, I would like to show you some data of our newest study, where we repeated the same paradigm with the puzzle solving task in parent-child dyads. I don't have any pictures of the fathers, sorry, in father-child dyads. I don't have any pictures of those yet, I'm sorry, but it was exactly the same paradigm. Mothers. And this paper is currently under review, but it is posted as a preprint. So if you're interested, please go and check it out on SciArchive. And again, this was led by Chin as a first author. We simplified it a little bit, so we only had 2 orders of cooperation and individual conditions, and always a rest in between. And we still looked at our 2 bilateral ROIs of dorsolateral prefrontal cortex and temporoparietal junction. And we were very happy that we could replicate the first finding, that interbrain coherence was again highest during cooperation as compared to both the individual condition and the resting condition. So we now have evidence for this from at least 3 independent studies in mother-child dyads, but also in father-child dyads. Interestingly, however, when we look at the father-child dyads in comparison to the mother-child dyads, we see that the highest synchrony was not necessarily in the same regions of the brain. So, as I have said before, the mother-child dyads showed the highest synchrony in the TPJ, whereas in the father-child dyads, this actually seemed to be the region where there was the least synchrony. So we seem to start to find some differences on the neural level in terms of where the coherence happens, even though overall the coherence pattern look very comparable. We then also found that in father-child dyads, there was a positive correlation between child agency and neural coherence during cooperation. So regardless of whether the children are interacting with mothers or fathers, it seems like the coherence is higher the more the children are actually allowed to take the lead and not having to follow the parents during this puzzle-solving task. And very interestingly, in this study, we for the first time found that individual differences in the view of fathers of how important they think that their role is as a parent were also correlated with interbrain synchrony during cooperation, in the sense that the more the fathers indicated that they think that they play an important role for their children as being a role model and as being a parent, the more synchrony there was during cooperation in these dyads. So again, we find that interbrain coherence relates to behavior so that it is actually functionally implicated. And we observed now some trait measures in fathers that were related to interbrain coherence. And again, this gives us some hints about relationship quality interacting with interbrain coherence and behavior, but this time in fathers and not in mothers interacting with their children. Now, this being said, we didn't find any links between coherence and measures of task performance and behavioral reciprocity in father-child pairs, as we did before in mother-child pairs, which points to the fact that even though we observe coherence, I'll be right done. Even though we observe coherence, the underlying mechanisms might actually be slightly different, and we will be very interested in looking into those differences. So to wrap up, I have shown that social neuroscience has recently moved from a first towards a second person approach, looking at direct social interaction in dyads, and that in doing so, we can look at the temporal alignment of biological and brain signals, which is summarized as bio-behavioral synchrony or interbrain coherence being one level of bio-behavioral synchrony. And we think that bio-behavioral synchrony is really important for close bonds and interactions, and this provides a direct link to attachment theory. The findings summarized in using FNIRS during parent-child interaction from an attachment perspective have shown that interbrain coherence is higher during cooperation as compared to individual task and resting, using at least two different paradigms. That inter-individual differences in behavior are associated with interbrain coherence, and there we have a range of behavioral measures that associate with interbrain coherence. Also, for example, free verbal conversation and turn-taking. We show that trait-like inter-individual differences are associated with brain coherence, for example, avoidant child attachment, maternal stress, role of the father. And this provides strong cues that the quality of the relationship or the quality of the attachment between parents and their children actually plays a role in interbrain coherence. And finally, there are some sex differences that I have been talking about, and there is a persistent interest in these sex differences, but we still need to have more data on that because it's not so conclusive at the moment. So the implications are that bio-behavioral synchrony and interbrain coherence could probably constitute new biomarkers for parent-child interaction quality and attachment. But we still have to see how bio-behavioral synchrony and interbrain coherence actually relate to other processes, for example, stress, and whether more coherence is always good or whether sometimes more coherence could actually even be bad. And as I said, there are some sex differences, but we still need to look more into those. So we are following up on this with still some studies in Leipzig, but now also hopefully starting new studies in Essex because I just moved here and we are setting up the FNIRS lab at the moment. So if you have any questions about that, if you're interested in becoming involved in this research, please do not hesitate to contact me. Check out my Essex website, my personal website, follow me on Twitter, get in touch. And thank you very much for your time and your attention.
Thank you, Dr. Vrticka. So there